from dual side construction. Thank you, Vince. And Vince is now sharing his presentation. Are you here, Vince?
Vince, please turn on your microphone.
Yes, your microphone is off.
Thank you.
Okay, now it's good. Thank you very much.
Sorry, I realized I had my microphone off and then couldn't find the controls to turn it back on.
No problem. Thank you.
Honeycomb construction has been the subject of my PhD working with Lars Chitka. And looking back at the history, honeycomb has been a fascination for some considerable time. A lot of the work has been looking at what it is that the bees build, but not so much about how they form it. So the form of honeycomb is a hexagonal prism with a rhombic structure at the base. The base is where I want to spend a lot of my time today talking about. The structure at the base comprises some fairly weird angles. If you measure what seems to be the natural way of measuring it, the lengths of the rhombus are strange. But if you measure it as dihedral angles, the angles at the base are all 120 degrees, which is, of course, the same angle that we see looking at the hexagonal section face on. So there seems to be a degree of commonality there. If you study honeycomb, one's attracted to study the regular nature of it. It is admirable in its construction, but of course, studying one regular hexagonal cell tells you no more than when you studied the previous one. And so what I've done is I've been spending my time looking at the imperfections, and we can see that they are able to build against irregular cavities in all directions. And this tells us a great deal about the means by which they build. And one of the things that is evident is that if you view it as a way of equal angles, both 90 degrees and 120 degrees are the same. And they build 90-degree comb at the edges across the top, or in this case, a stimulus that I gave them. And this is the beginnings of some idea as to how they build the bases. The methods that I used to construct, to do my research, is by photographing a sequence of stimuli that I gave them before they went in the hive, after it came out of the hive, then overlay the photographs, but by getting common reference points on both photographs, I was then able to align them to compare the results with the stimulus directly. The first part of my work looked at the face of the honeycomb, and it was clear that there was a degree of cell wall plasticity in that if I gave them a straight stimulus, it just vanished once they had finished constructing the cells. And so this talk is looking at the plasticity concerned with the base construction. The first experiment that I did was to offer them a dimpled piece of wax, but with a barrier preventing them access to the backside of the wax. And the prediction was that they would focus their attention on the raised parts, turn that into honeycomb, but the bases of the comb would be domed until I then subsequently gave them access to the back, at which point that would then proceed into full faceted bases. I wanted a non-destructive mechanism for measuring it, so I fabricated a profile gauge using hypodermic needles and thin wire. And so I was able to then offer them such a stimulus, and we can see that the first stage, it was domed bases that they formed, curved bases. The picture in the middle at the bottom shows the reverse side, where the dome is actually raised above the level of the wax. But then once I removed the barrier from the reverse side, the very same cell had then progressed to become faceted. Using the profile gauge to measure the shape at the base of the cells, both before and after, and indeed the domed side at the back, I was able to get some statistics as to how well the profile of the base fitted the curve or faceted, and a proportion of whether it looked more curved than faceted. And so here we see the population having been given access to the one side, and then the second group is those same cells having been back in the hive for a second occurrence, but still restricted to one side. The blue is what was curved, but with the barrier removed, became faceted. And for reference, the yellow is the profiles that I measured on natural comb, where they were allowed access to just build it freely. My second experiment was to offer them a thick piece of backing wax, and my prediction was that because they were unable to get to both sides of a common wall, both sides would start curved until they were eventually eroded to the point where the efforts would meet, and then they would become faceted. And indeed, that is exactly the characteristics that were measured. So the large red box plot shows the curved at stage 1, and then sometime later, when the cells had progressed to approximately 8 to 10 millimeters deep, the bases became faceted. And for comparison, you can look at the similar split that I achieved by preventing access to the back of the wax at all. Bees are known to build optimum forms of honeycomb cells. Well, in fact, they will just go and build whatever they're able to build, and I coerced them into building the worst possible form by offering them a stimulus where I made the dimples directly opposite each other, and as a result, they then worked on cells both sides of the wax stimulus until they encountered each other at the middle, and proceeded to flatten the wax at that point, but it was a single flat surface. And so the bees had just followed their natural inclination to maximize the space, but directly opposing, they maximized it to the point of being the worst as far as storage is concerned. The cells still worked. As you can see in the picture, the queen has quite happily laid eggs in the middle of what were flat cells. So in summary, the work that I've done, both looking at face-on appearance of the hexagons and now the bases of hexagons, the bee's natural inclination is to form a spherical start to the cell by circular and spherical motion of the head. She lays down wax and sculpts it to a spherical shape. That sphere becomes a circular tube as the tube is lengthened. And in an isolated cell, the goal to maximize the volume of the cell will cause all of those sides to be curved, both the walls and the base. But in the earlier chapters of my work, the face-on hexagons, when side by side, give a flat face in the junction. When you get a three-way intersection, the mutual effort on all three cells being worked on simultaneously pushes the wax around to form the commonly recognized 120 degrees. But here we see that at the bases, when you see the four opposing cells that happen when cells from one side are in contact with the cells on the other side, the mutual effort from all four cells arrives at the spherical coordination of 120 degrees. So I would like to thank Lars for supporting me during this work, and my audience here for listening to me. Thank you very much, and my acute apologies for the mistakes at the beginning.